Now we are going to add another speaker to this session for future systems. That is Sandeep Kulkarni, the founder and CEO ImageProVision Technology. Sanjay is going to talk to us about artificial intelligence-based particle characterization complying to regulatory requirement. I'm not going to read all his resume, but it is important to know that he is an avid enthusiast in field of training, teaching. He always finds opportunities to interact with people and extend his experience and learn from them. This is something which is very good about Kulkarni. Sandeep, please take your seat.
Thank you very much. Am I audible? Yes, okay. Thank you for the opportunity IDMA has given for a change, a little bit of different subject, which I'm going to talk about. It's artificial intelligence-based particle characterization and how it can help in regulatory requirements. So there are two innovations I would be talking, and both the innovations is happening in here, right here in India. So one is, of course, particle characterization. We'll just, all of you are experts in this, so I don't have to tell much about it, but we'll just go through what is particle characterization. And then the first innovation, what I'm going to talk about is the microscopic particles characterization or particle size and shape analysis, how we do it. And then how we integrated the artificial intelligence. I mean, I was in Hyderabad yesterday in the BioAsia conference, and that is the buzzword. Everyone is talking about machine learning and artificial intelligence. But how we have actually implemented it, that little bit about it I'll tell. And then how everything goes into a cloud platform, and then we leverage the efficiency of technology in solving some of the quality and the regulatory issues what we face. So why particle properties are important, you all know. Just I've listed down few of them. Better control on product quality, improves product performances, troubleshoot manufacturing and supply issues. Many a times I come across many of problem statements that the moment I change a supplier, the performance of the drug batches are changed. However, the input parameters look pretty similar. So what are those? So there are certain things which we can work out in this space also. Better understanding on products, ingredients, processes, optimization of efficiencies, manufacturing processes, yield improvement, and of course, state of the competition. Again, role of particle characterization, we all know in pharmaceutical industry, particle size, particle shape, particle size distribution of active pharmaceutical ingredients known to strongly affect the stability and aesthetics of drug formulations. The size and shape of particles used in pharmaceutical product can impact the dissolution rate, influence the solubility, addition, and dispersion of particles. I just give one example how this affect actually is there are two types of batches we compared here. The dissolution profile of one of the product API, which has got three different particle sizes like 128 micron, 135 micron, and 120 micron. Not major difference. The dissolution profile is pretty much same. I'm not getting this. So pretty much same, the top one. But the two batches with SR granules, one with 675 micron particle size, and the other one is 625 micron particle size. So you will see that red one has got a better dissolution profile because of lower particle size. So I'm just trying to correlate build a case how this affects, the dissolution profile. So whenever we are talking about the drug efficacy or drug efficiency, the dissolution comes as one of the important parameter. So this is a simple equation of rate of dissolution, which is directly proportional to surface area. And there are other parameters as well, but I'm just concentrating on the surface area. So the traditional way of doing this correlation is with the particle size. So, I mean, a very simple illustration. This is a one particle with this volume. If I cut it in between, the volume or the mass remains same, but I have generated two more surface area. So for the same volume, you have more surface area, and that's why more dissolution profiling. So as you keep on reducing the particle size, the surface area changes, and that's how the dissolution profile also changes. And to what extent? It's a very interesting slide. For a perfectly spherical silica solid particle, if you take particle size of 1,000 microns, the size of the surface area of 1 kg of this is almost 10 square meter. But as you keep on reducing the size, if you come to 0.1, the surface area is as good as a football field. And if you reduce it further to 0.01 micron, it is as good as the surface area of a Disneyland. So for the same material, if you're reducing the particle size, the surface area changes, and that's how the behavior or the performance changes. But this is not the only story, and that's why most of the people were trying to correlate the particle characteristics only with the particle size. But with our innovation, what we have found is that along with particle size, particle shape plays a major role. Now here I'm just giving 4 different types of shapes of particle. Now what we have done is that if you keep the volume of this particle same, that is 523.81 micron cube, in a traditional way of doing particle analysis, that is laser diffraction technology, which takes the image and converts into an equivalent sphere, it will give the diameter of that equivalent sphere 10 micron for all the particles. But if you look at the surface area, for the spherical particle, of course, it's the minimum one. But as you are changing the shape, for a cube, it will be 24% more than spherical. And in the last case, where it's a cuboid with 4:1:1 size of sides, your surface area increases 45%. So it's very important that along with particle size, you also should know how particle shape is. So is there a method by which you can do very quickly the particle shape? So this is where we have innovated a way of doing the particle size and shape characterization using a microscope. What we have done is, any kind of microscope, irrespective of make or model of microscope, you can put it, connect it through a camera to this software. By this, what happens is, whatever sample you are doing under the microscope, everything goes to the software. The algorithms are such that it identifies every particle, and then all data goes into the system. You reduce particle analysis time almost by 50%. And of course, the last one is eliminate the inefficient and error-prone, tedious data processing work. Along with that, the other innovation which we had done is that in the same analysis, using the optical parameters like particle size, shape, color, texture, intensity, you are able to differentiate different particles. Like in a formulation, you will be able to know how much API. If you have multiple APIs, you will be able to know the size and shape of multiple APIs. So algorithm, I'll quickly tell about it. The top images, that's how you get the microscopic image, and that's the input image. What software does is that, first of all, it identifies each and every particle. Here we are using some part of machine learning where we are trying to identify, I will talk a little bit more in the next slide, but this is where we do first type of classification where whether it's a isolated particle or an agglomeration. So agglomeration is a reality, we can't avoid it because it's the particles coming together forms an agglomeration. So first classification it does is that it's isolated particle and agglomeration, and then gives the statistics separately for isolated particles and separately for agglomeration. And within those isolated particles, using this machine learning modules, we are able to differentiate particles in terms of API, excipients, or multiple APIs. Now, again, size we talked, we all know if my particle is near spherical particle, the diameter of the particle would be the size of my particle. But if my particle is irregular particle, so we define the shape in multiple ways. That is ferret's maximum diameter, ferret's minimum diameter, circular equilibrium diameter, circularity. So all these data is populated, and that's what you get in the report. So you run a simple your analysis, sample analysis on any kind of microscope. If it is connected with this platform, you will get all these, each and every particle, how it looks and different parameters of it. Now, how does it help? Because this microscope, which is still considered to be a manual instrument, the result of the microscope is as good as the person who is doing it, has the whole subjectivity is removed. So we talked about automation. This is one kind of automation, but automation is where you are removing some of the physical activities of a human and repeated activity which you automated. Once we go into the artificial intelligence, actually what we are doing is, along with the automation, we are putting some intelligence into the system. Just to give a small idea about it, right? If you have a group of people, out of which one is the most intelligent person, if we put the way he thinks or the way he takes the decision, the way he interprets the data, that into the system, then it becomes an intelligent system. And now if any other person is using the system, the result will be as good as the most intelligent person is giving. So that's how it works. But with this system, since your microscope is connected to this platform, you have got secured access, you have got password validity, a detailed audited since logging in till the logging out. Whatever activity is doing, everything is recorded. So I just touched about the machine learning and artificial intelligence part. It's a very little complex subject, but I'll just try to give in 5 different steps. So how it works, especially in the, see, a lot of people have done the machine learning or artificial intelligence use of this in data, data mining, understanding the data, but what we have done is we have used it for the images. So first machine learning engine works on the image quality. So as you are taking multiple images, this machine learning will identify whether this image is correct for analysis or not. Sometimes the light condition is not good, or if a user has made a very clumsy sample with a lot of agglomerations, or there is no particle at all. So there is some decision point here which this machine learning engine works on it, and then it qualifies the image for the pre-processing. Then pre-processing, once you're done, then second machine learning engine works where it is doing the particle identification and particle classification. Now again, there are two ways of doing it. We are doing, and let me tell you, artificial intelligence is not the human brain, you know. It's just augmenting the human intelligence. It's not replacing the human intelligence. You need to have a human intelligence into it. So what we are trying to do is, through automation, we are saving the time of a person. Through artificial intelligence, we are saving the decision-making time of a person. So we are trying to give some indication to him to take certain decisions. So in this machine learning classification engine, all the particles what we have captured, they go and classified as APIs or excipients. So if you have multiple particles into it, and you want to bunch it in a separate compartment, like I have 100 particles, 10 of those are of this type, 20 of those are this type. Machine learning will give you those. But what is this 10 calls? That is where human intervention comes. And all this then data goes into the artificial intelligence engine through various algorithms. And as you are using the system again and again, it becomes more and more intelligent, and you get more authentic data. So these are some of the screens I have not get into. So what happens is, first of all, you have to train the system with some of the basic data. Like if you have a certain kind of APIs, you run the pure API sample, this system will take all the images of API particles and train itself. Similarly, you do for different, going forward, what is going to happen that all API companies, they will put the particle characterization on this platform. Excipient companies, they will put their particle characterization on this platform. The formulation guys will pick up and use this and do the predictive analytics. Because now I showed you how it is related to the dissolution profile. So all this, if you correlate, it gives a huge amount of benefit to the industry. So there are various different types of statistics which are published in this, like how many are APIs or how many are excipients and agglomerations and other details. So D10, D50, D90 graph is plotted, which gives the indication that how your sample is homogeneous or not. So all this data is populated. Here you can see each and every particle like I showed, and details of every particle you can get here. And this all happens, let me tell you, in few seconds. So that's the power of technology. This is our new innovation, which is called MorphoViz. It's a cloud platform where what is going to happen is that every company, I've seen that most big companies have got hundreds of microscopes. Some of them are in microbiology department, some of them on formulation, some in analytical QA, QC. So what we are going to do is that you can connect all these microscopes in a single platform. Now this cloud platform is available as a public cloud as well as a private cloud. And also it's a cloud agnostic. So you can put it on Microsoft or Google, or you can put it on Amazon, any cloud it works on. So what will happen is that, like if you want to do PSD, particle size distribution and particle characterization, let's say it goes into analytical department, they can use it. The other one is API excipient classification. The third one is like injectable plants or ophthalmic or inhaler plants. They do USP 788, 789, particulate matter count. It goes into that. Hot stage microscopy, if you want to see the particle behavior in terms of time and temperature, it does that. Nanoparticles, you have nanoparticles, you do the particle. If you have nanoparticle SEM, TM images, you can just upload and you can get all the details. Also the microbial analysis for gram staining positive, colony counting, everything, whatever you do with particle, with microscope, it can be done. And most of is that it's a 21 CFR or any European standard compliant. So this all does this. So this is what I was talking about. I hope this makes some interest. Thank you.
Thank you, Sandeep Kulkarni. It's a great knowledge to all of us. I must tell you that there are APIs where you require to slow down the dissolution by increasing the particle size of the. I don't. Yes, Milind, you can take over.